Hey everyone, Mr. Gibson here. So I wanted to record this video to kind of walk you through what I'm expecting for Explore 2 Part B for Make Me a Sandwich. So up till now, you've looked at that analogy of how a sandwich was made. You've connected all of the different parts of that analogy to the actual parts of protein synthesis inside of the cell. And you've put the function of all of those parts, and you answered some questions, and you were able to turn that into me. And at that point, whatever you had down was okay. Hopefully, the video and any other resource you were able to use was able to help you get that correct. But I didn't want to just leave you hanging, hoping that the analogies that you made, the connections that you made were absolutely correct. I wanted to make sure that we were all good to go and on the same page before moving on to Explore 3. So here is what you're going to do for this activity. It's pretty straightforward. It says, now that you've made an analogy to explain the parts of protein synthesis, I want to make sure we're all on the same page. So you're going to watch the attached video, which is the one I'm recording right now. Then you're going to open the Google slide file and copy and paste the slide with the correctly filled in chart into your interactive notebook. So let's take a look at this. Here's the instructions again. Here's the chart. So the house represented the nucleus. Okay, so you had the boy who broke his leg or whatever, he's injured and he's stuck in the house. And just like DNA is stuck inside of the nucleus, the DNA cannot leave the nucleus. It's too large of a molecule, and you wouldn't want it to leave the nucleus in the first place because it has all the genetic information for how all of your cells are supposed to function. You don't want to accidentally lose that somewhere. So it needs to stay in the nucleus. So the boy stuck in the house represented your DNA because it's stuck in the nucleus, and the house represented the nucleus. And again, the nucleus, it contains your DNA. It contains all your genetic information. And DNA it holds all of the information, all of the instructions for how to make all your proteins. It's almost like a cookbook that has all the different recipes for how to make all the different proteins, okay? So the boy who wanted the sandwich, he had the instructions for how he wanted the sandwich to be made. So he represents the DNA here. Now, since the DNA can't leave the nucleus, how does the instructions for how to make proteins get out of the nucleus? Because proteins aren't made in the nucleus, they're made outside of the nucleus. Well, we have to have this molecule that will carry the message from the DNA out of the nucleus for us. And that molecule is called mRNA, okay? RNA stands for ribonucleic acid. DNA stands for deoxyribonucleic acid, so they're kind of cousins here. The M in front of RNA stands for messenger. And what this is it's a molecule similar to DNA, but it has some important differences. One, it's really short, okay? Much shorter than DNA. Your DNA is long enough that if you were to take all of your DNA and stretch it end on end, it would reach from here to the sun and back 6 times. So pretty long. But your RNA is relatively short because it needs to fit out of the nucleus. So the M tells us its function. It's a messenger. It's just carrying a message. So what it does is it goes to the DNA, finds the gene, finds the recipe for exactly what protein needs to be made, copies that down, and then takes that message out of the nucleus. So it moves the code from the nucleus to the cytoplasm to make proteins, okay? Now, our sandwich builder, the person who is making the sandwich, or in real life, we're looking at what makes the proteins. Well, that would be the ribosome. The ribosome is probably a word you haven't heard before, but that is an organelle, or it's a structure inside of your cells that makes proteins. It's like a protein factory, okay? So the mRNA goes to the ribosome to tell the ribosome how to make the protein. Just like the sibling went to the sandwich builder to tell the sandwich builder how to make the sandwich, the correct sandwich. Okay. I'm going to move on from this. I'm going to come back to the sandwich shop owner. How does the ribosome get the different ingredients it needs to make the proteins? Well, there are these other types of RNA floating around inside of your cell, inside of the cytoplasm in your cell. One of them is called tRNA, and that's T stands for transfer. So it's transfer RNA. And its job is to take all the ingredients to make the proteins that the ribosome needs and bring it to the ribosome. Just like the ingredient carriers, like the sandwich shop or the sandwich builder had to go get the ingredients from the ingredients carriers, the ribosome has to get the building blocks for the proteins from the tRNA. So the tRNA will actually move into the ribosome, drop those off, and then leave. Okay, those ingredients, those building blocks that make proteins are called amino acids, and we're going to learn more about those in a later unit. But right now, you just need to know that amino acids are the building blocks of proteins. Okay, now the finished product, once all the amino acids are assembled, once all the ingredients were put together in the correct order based on the message carried from the nucleus, we call that finished product a protein. Where in the analogy, it was a sandwich. And that is the end product of all of protein synthesis. That's the whole point. That protein then goes on to give our cells the specific traits that they need, the specific characteristics they need to do their job wherever they're located in the body. Now, some of those proteins need to be packaged and sent out of the cell, and the structure inside the cell that does the packaging is the rough ER. It stands for endoplasmic reticulum. It's okay if you just know it as rough ER for right now. So the rough ER will package up the proteins and send them where they need to go. Just like in the analogy, the sandwich shop owner is the one who packaged up the sandwich and sent it where it needed to go. Okay, so I hope all that makes sense. I hope that analogy helps this otherwise really abstract concept really make sense. So that's just the first part of this assignment is I would like you to read it, make sure you have those right, and then copy and paste that into your interactive notebook. Now, the second part is you're going to go here and you're actually going to do a practice CER. You guys did a pretty good job on your first CER on the quiz, but I want to make sure we're good to go on CER, so we're going to do a practice one. So this is Explore 2B CER, and here's the scenario for your CER. So now that you understand the general steps of how proteins are made, you got DNA being transcribed or being copied into RNA, and then that RNA is then translated by the ribosome into a protein. So you got transcription and translation going on. So now you've explored that. What if something went wrong? What if this process of protein synthesis, either in transcription or in translation, a mistake was made? Okay, so we call that a mutation. Mutation is when there's a miscoding or a misreading, a miscopying down of the instructions in the DNA sequence. Okay, they can sometimes, they don't always, sometimes you can have silent mutations, meaning you don't ever notice them, but sometimes we can have a mutation that causes a protein to be built incorrectly. One disease that causes this that's caused by this is sickle cell anemia. You may have heard of this. This occurs when the gene that has the instructions to make hemoglobin, which is the protein in your red blood cells that carries the oxygen, that something goes wrong when that gene is being copied. Okay, so that causes hemoglobin to be built incorrectly. Okay, think about it. If the sibling had written down the instructions for how to make the sandwich wrong, they would not have gotten the correct sandwich. Same thing here with hemoglobin. The instructions are messed up, hemoglobin doesn't get made correctly, and what happens is that causes the red blood cell, instead of being this nice normal round red blood cell, they are all deformed and sickle shaped, and so they can't function properly. They can't properly transport oxygen through your body. So what I want you to do is answer the CER. How does a mutation that changes DNA cause a protein to be built incorrectly? Where is that relationship between DNA and protein? Why does changing the DNA affect the protein? That's basically what you're trying to answer here with a claim, some evidence, and some reasoning. Okay, if you guys have any questions, feel free to ask me, and good luck.